Hello everyone, welcome to my final lab presentation video. Today I'll be presenting lab number 4, Lactase enzymatic activity. In the lecture, we've already learned that enzymes are remarkable biological catalysts that increase reaction rates by at least a millionfold. In this lab, we specifically focused on lactase, a glycosidic hydrolase that catalyzes the breakdown of lactose into glucose and galactose. This enzyme plays a critical role in mammalian digestion and has significant medical relevance. So in this video, I will take you through the background science, our experimental process, and also the real-world applications of our findings. Let's first look at the background information. Enzymes function as biological catalysts by lowering activation energy barriers, allowing chemical reactions to proceed under physiological conditions without requiring extreme temperatures or harsh chemicals that would damage cellular structures. They are recognized as highly specific protein molecules, each designed to catalyze particular chemical reactions by binding to specific substrates at their active sites. And the thermodynamic principles underlying enzyme function are also fascinating. As I just mentioned, enzymes operate by lowering the activation energy instead of changing the overall free energy change of the reaction. This activation energy is actually the initial energy needed to start a chemical reaction. As this picture illustrates, enzymes create an alternate reaction pathway with a lower energy barrier, dramatically increasing reaction rates without being consumed in the process. And specifically for this lab session, we focus on lactase. Lactase belongs to the beta-glycosidase family of enzymes, a highly conserved group present from E. coli through to humans. The enzyme exists as a tetramer with 4 identical subunits working together to create the active complex. This tetrameric structure is essential for proper function. Each monomer contains an active site where the substrate binds and where catalysis occurs. The specificity of lactase for a substrate is determined by the precise three-dimensional arrangement of amino acids in its active site. Then let's look at the mechanism of lactase reactions. Lactase catalyzes the hydrolysis of the beta-glycosidic bonds in lactose, cleaving this disaccharide into its component monosaccharides, galactose and glucose. These simple sugars are then utilized by cells in metabolic pathways to generate ATP. During infancy, lactase is highly expressed in human digestive system to break down lactose in breast milk, which is a key source of nutrition for developing infants. So we conducted an experiment to measure lactase activity under varying conditions. Since neither lactose nor its breakdown products absorb light in the vertical spectrum, we needed a clever approach to quantify enzyme activity spectrophotometrically. To solve this detection challenge, we used OMPG, which mimics the structure of lactose. When hydrolyzed by lactase, OMPG releases orthonitrophenol, which has a distinctive yellow color that can be measured using a spectrophotometer. This approach allowed us to quantify enzyme activity by measuring the accumulation of this colored product over time. So we began our experiment by extracting lactase from commercial dietary supplement tablets. This involved crushing a lactase tablet containing 200 mg of enzyme into a powder, suspending it in phosphate buffer saline, and centrifuging to remove solid particles. The resulting supernatant contained our active lactase enzyme for use in subsequent reactions. And the core of our experiment involved investigating how temperature and time affects enzyme activity. We conducted reactions under 37 degrees, which is the optimal temperature for human lactase. And we established a series of reactions that were stopped at different time intervals, including 30, 60, 90, and 120 seconds. By stopping the reactions at precise times using sodium carbonate, we could plot how product formation changed over time, effectively measuring the rate of the enzyme-catalyzed reaction. After processing all our reaction tubes, we transferred the solutions to a 96 well plate and measured absorbance at 450 nanometers using a microplate reader. We then plotted absorbance against time. This allowed us to calculate the rate of production formation, which directly correlates with enzyme activity. As expected, we observed that reaction rates are high at 37 degrees, confirming the temperature dependence and also the predictable kinetic patterns of enzyme reactions. Then let's look at some real-world connections. The medical significance of lactase extends far beyond our lab experiment. Lactase deficiency affects approximately 65% of the human population, leading to the condition known as lactose intolerance. This occurs when individuals lack sufficient lactase enzyme activity to digest lactose in dairy products. Without adequate lactase, lactose passes through undigested into the colon, where gut bacteria fermented, producing gases and exes that cause the uncomfortable symptoms associated with lactose intolerance. Thus, the experiment also has substantial commercial applications. Lactase supplements enable lactose-intolerant individuals to consume dairy products without discomfort. Additionally, the food industry uses lactase to produce lactose-free milk and also other dairy products. The enzyme's ability to break down lactose into glucose and galactose also improves the sweetness and texture of dairy products, as these monosaccharides have greater sweetening power than lactose itself. And at the end of this video, I would like to express my most sincere gratitude to those who have made this academic journey both possible and meaningful. Firstly, to Professor Zhuhun Li, I really appreciate your guidance throughout this course. Your passion for the class and biology knowledge has been so infectious, inspiring me to delve deeper into understanding the biological systems. Thank you for designing many thoughtful sections like Think Per Share that bridges theoretical concepts with practical applications. Your patience and care for every student touches me a lot during the individual meetings in lab sessions. I am particularly grateful for your encouragement and your willingness to answer my numerous questions after class. Thank you for being such an important part of my biology journey so far. And also to my incredible Think Per Share team members, Fei Fei, MJ, and Yanjia. This class wouldn't have been so enriching without your collaboration. Your diverse perspectives and also approaches to problem solving have expanded my understanding in ways I could not have achieved alone. I hope our paths cross again in future courses. Thanks a lot for watching my video. See you in the next lab.